Okay, so I'm going to go through some of worksheet 7 number 3. Specifically, it's in relation to Benzer's experiments, and students are having trouble understanding the difference between B and K bacteria and how many plaques are going to be on each. So I want to draw out what's happening here. So first, let's just go over some basic things. So the first thing is understanding the difference between B and K. So let's talk about B bacteria versus K bacteria. So the first thing is that B is what they call permissive. So all we're saying is that both wild type and mutants can lyse this bacteria. Okay, this is different. Lysing is different than infecting because infecting can happen on both bacteria. It's just whether or not the phage can infect them. Or can lyse them. Ugh, of course. So it's it's whether or not they can lyse them. K can be lysed by wild types, and not mutants alone, but they can be lysed if you have complemented mutants. Okay, so the first key thing is that B can be lysed by wild type mutants, and this mutants can also be complemented mutants, it doesn't matter. K can only be lysed by wild type or complemented mutants. So that's the first key difference between them. The second key difference is what type of event, genetic event, we're allowing to occur in each. So when we're talking about B bacteria, we're specifically asking the question of, can we have a recombination event? We're only interested in recombination in B, and in K, we're only interested in complementation. You know, this one is easy to remember because C and K have a similar sound, so C and K complementation, that's what is allowed. So we're only looking at recombination and complementation. So when we have these types of problems, first you have to distinguish between your types of bacteria. The next thing is figuring out whether or not your mutants can recombine, complement, both, or neither. So the way to do that is by drawing out the genes. So specifically, we're going to be looking at worksheet 7 number 3, and we'll be drawing out those genes specifically. So say we have, let me just get some colors. So say we have our A and B gene, and it tells us that mutants 1 and 2 are in A, and it tells us that 1 is a point mutation, 2 is a deletion. So I'm going to skip over how to draw this out because ultimately it's just using the information they give you to figure out how the mutations are aligned. So what I'm going to do is put 5 overlapping both, it's a deletion. Then I'm going to put a point mutation here, which is 4, which overlaps 5. 3 does not overlap either. 2 overlaps 5, but we don't know where 1 is, but it is a point mutation. So the best way to do that is to have this parentheses and then to have an open piece on this side of 2. So that's telling us we don't know whether or not 1 and 2 are overlapping. So now we just need to review some basic ideas. So complementation, we talk about complementation when you have two genes, and from one genome you have a good A but a bad B, and from the other genome you have a bad A but a good B. So when you're complementing, that means that your mutants are in different genes. Okay, so that's complementation. Recombination recombination can occur as long as you're not overlapping. So if we're thinking about what this looks like, let's say we have this gene, or this strand, and this strand. And this one, we have a mutation here, and we have a mutation here. So here, we can have a recombination event because we can go like this, and we can have a crossover in between here where we'll end up with two nice pieces. So that's recombination. So you'll have recombination if mutants do not overlap. Okay, and then just for completion, let's just look at what complementation is saying. So this is what we're looking at. Say we still have this same mutation, but now the question is, if this is gene A and this is gene B, can they complement? And the answer is yes, because here on the blue strand we're saying we have a bad A but a good B, and here on the pink strand we're saying we have a bad B but a good A. And so if these two are together in the same bacteria, we'll have a good A and a good B, which is what you need in order to lyse the cell. So that's the second piece, is understanding whether or not you can complement and recombine. So when I go through these problems, the first thing I always do is draw out what my mutation map looks like. That's not a technical term, it's just understanding where things are. The second thing is to go through each pair that it gives you in the cross and to determine if it can complement or recombine. So let's look at what it's going through in part A. Okay, so we didn't even look at what the question is asking us yet. We're just looking at the different crosses. So if we, sorry, not trying to move this out of the camera. Okay, so if we look at crosses 1 and 2, 1 and 3, 2 and 4, let's just start with the first the first 3 rows. So what we need to know is, can they complement, can they recombine? Okay, so going back over here, things complement if they're in different genes. Are 1 and 2 in different genes? No. We know they're both in A, we just don't know if they overlap. So we know they cannot complement. Can 1 and 2 recombine? Do they overlap? We don't know. So I'm going to put a question mark here because we don't know if they can recombine. Now what about 1 and 3? 1 and 3 in A versus B. That means, yeah, they can complement. Can they recombine? Are they overlapping? Answer is no, they're not overlapping. So yes, they'll be able to recombine. How about 2 and 4? Same thing. You'll be able to complement because you're in different genes, and they're in different genes and they're not overlapping, so they can recombine. So that's this next part. 1, draw out your map. 2, figure out whether or not things can complement and recombine. Now the last thing is understanding what happens when you're plating things on B versus K. And this is where high and low MOI become important. So when we're talking about high MOI, what we're saying is you have more phage than bacteria. So you have greater than 1 phage infecting 1 cell. And as soon as you understand what that's telling you, then you'll be able to understand what's happening. So now, say we have 2 phages, and I'm going to use pink and orange as my 2 different phages. So we have this phage infecting one cell, and then we also have orange phage infecting this cell. And so you have two phage infecting any one bacteria, which is good because that is what will allow you to either have recombination or complementation. So now, because we have more than one phage in any one bacteria, we're allowing both the purple genome and the orange genome to come inside the bacteria. So if they can complement, they'll complement. If they can recombine, they can recombine. So that is what we're doing here. When we say high MOI, we're just saying that we have two phages that can infect one bacteria. Oops, wrong color. Okay, so then if we go low MOI, that's just the opposite then. So you have less phage than bacteria. So that means you have one phage infecting a cell, or maybe even less. So that means anytime you have a phage infecting, it's only going to infect one bacteria, which means you can never have a recombination or complementation event because you only have one type of DNA inside the cell. So this allows complementation or recombination. And remember that whether or not you're complementing or recombining is dependent upon which bacteria you have this high concentration on. Because B, we're only talking about recombination, and in K, we're only talking about complementation. So if you're a low MOI, this means you will not have any complementation or recombination. Okay? So this is the difference between high and low MOI. So when you're going through these problems, the first key thing, figure out what your mutant looks like. Second, figure out whether or not these mutants can complement and recombine. That was this part. And now the third part is actually going through and understanding what is happening in your experiment, because now the order matters. And the order matters because we care about our lysate in each part, and that is where you need to understand what's happening. So in part A of number 3, it tells us that we have a high MOI infection on B. So we have, I'm just going to abbreviate that like this. We have high on B, then we take this lysate and take that and put it high on K, and then we get lysate out. And we go low on B, low on K. And the question is, what are we going to see in terms of our plaque number? So what you have to do at each step now in phase 3 or in part 3 of this question is figure out what is in your lysate. So the key thing here is understanding what's in your lysate and how that's going to infect. So let's work with specifically this one when we have 1 and 3. Let's work with this one and see what happens in our cross, or in our experiment. So we start with 1 and 3. 1 and 3 we said can complement and can recombine. So if we're plating that at a high concentration on B, then we're interested in whether or not by plating 1 and 3 on B at a high concentration, will we allow recombination? The answer is yes, because we already said 1 and 3 will allow recombination. So that means we'll allow recombination, so we will get wild type mutants out. So we'll get wild type we'll get mutants, and we'll get the like super mutants. So we'll get like the negative mutants. So when we allow recombination to happen, like we said over here, basically what we're getting, let's go back to what we're getting in terms of our pink and orange phage, because that's what we're working with. So if we have our 1 and 3 mutant, which 1 is a point mutation, 3 is a point mutation, that makes it nice and easy. So this is what we have. And then what we're saying is we have a complementation event occurring, recombination event occurring. So we'll get this one, and you'll get this one, which means that in our phage that we get out, our lysate will have phage that are wild type, because we had this piece, so we had this good piece, so that's a little piece of the orange here and a little piece of the pink here, so we'll get wild type. If a recombination event does not occur, that means we'll just get our mutants. So if we don't have a recombination event, we'll get our pink or we'll get our orange, both with their original pieces that are messed up. And then if we get the other type of recombinant, then we'll get both the purple and the orange. But now this one is going to be the minus mutant, where we have both pieces like that. So if we can have recombination and we plate high on B, what's going to be in our lysate is wild type. So we have mutant, that's pink, we have mutant, that's orange, and then we have a double negative mutant, so negative mutant. So those are the things that we actually get in this lysate. And now we take this lysate, and that is what goes in plates on K at a high concentration. So you have to understand what is happening at each step in order to understand what is coming out of your lysate, and then if you go and plate that lysate on another bacteria, what is going to happen. So here, what we did is we said, if we have 1 and 3, we know that they can complement and recombine. If we plate it at a high MOI on B, that means we're allowing recombination to happen. Because we already established that recombination can happen between 1 and 3, that's telling us that we'll be able to get wild types from having a recombination event. We'll get both the mutants out if a recombination event doesn't happen, and then we'll get double negative mutants if we get the crossover recombination event that gets both bad copies. So that's what's in our lysate now. And so now this lysate is going and getting plated high on K. So here we have to say, okay, we're plating high on K, which means we're allowing complementation to occur. So the question is, can we complement with what is in our lysate? And so what we're looking at is, can these things complement? So we already said that 1 and 3 can complement because we looked at them and they're in different genes. So they can complement, and if they complement, that means they'll be able to lyse K. The key thing is, even when you have mutants that can complement and lyse K, that doesn't mean you're going to get a wild type out from that specific lyse. Because just because you're complementing, you're never exchanging DNA, so you will still get the mutants out from just that complementation. With that said, we still are plating wild type on this plate, which means the wild type will infect and lyse K perfectly fine. So that means in our lysate, what we're going to have is basically the same thing here. We're going to have wild type going to have our wild type phage, and we're going to have our mutant phages. So finally, we get this next piece from our lysate, and that is now what we're plating low on B, low on K. So now we have wild type and mutant phages, and now we're low on B. So B can be lysed with wild type or mutants, which means both our wild type and our mutants are going to be lysing the bacteria. So remember, our mutants were both orange and pink. That's not orange, that's yellow. They're both orange and pink. And we just said our wild type was, let's just call it blue. So that means on this plate, low on B is going to correspond to a plate that has wild type plaques, has mutant plaques of both colors, and these will all be able to lyse B. But now, when we go and plate this on K, because we're a low concentration, that means the only thing that's going to be able to lyse it is the wild type. So that means the only plaques that you're going to see here are from wild type. So then comparing the difference between the plaques between B and K, that tells us that you're going to have more on B because your lysate had wild type and mutant phages. And so when you have this type of setup where you're ending on a low plating, then if you have both wild type and mutants in here, then you're going to have more on B. Okay, so let me just very briefly bring out a different page, and let's just review. Yeah, I don't want to write on the back. Let's just review our process for what we did. So first thing that you want to do is you want to map out the mutations. Okay, so we mapped out the mutations because we needed to see whether or not things overlapped, if they were in the same gene, etc. Because this will allow us to know, can mutants complement? Can mutants recombine? So that was what we were answering in the first step. We were figuring out, okay, based on what these mutations look like, we need to know, can these things complement? And so this was really what we were calling step 2, because first map it out, then figure out what that tells you. And then the third step to this was really understanding what was happening at each step in the experiment. So this was, how do we say this in a simple way? I guess honestly, it's saying map out experiment. So let me just go into more of what that means. So mapping out experiment, really what we're saying is if we go high on K, then we go high on B, then we go low on B, low on K, versus if we go high on B, high on K, then low on B, low on K, which is just what the question in worksheet 7 was doing. Then what you can say is, okay, in this stage, high on K, we're looking at complementation. This stage, we're looking at recombination. this stage, because it's just low on both, you're really looking at, do you have wild type or wild type plus mutants in your lysate? Because that's really going to tell you, or nothing actually, that's another option. That's going to tell you how these two bacterial lawns are going to differ. And so then you can say the same thing, but in the reverse order here. So if you start on B, then you're first starting with a recombination event. Then you're going to look at whether or not complementation can occur. And then the same thing that we said above, once you go to low B, low K. And you have to be careful because not all of the questions are the same. They're not always going high, high to low. Sometimes it's just high to low, sometimes it's low to high, so you really just have to look at what's happening in each step and figure out what you're allowing to occur and whether or not that can happen based on number 2. And so the key thing here with mapping out the experiment, there's like a part A, which is looking at complementation, recombination, what's happening at each step. But then the part B to this is understanding what's in your lysate. Because what's in your lysate is going into the next part of your experiment, and that is really the key thing to understand. What is in the lysate? So the final thing that I'm just going to go over really quick is what happens when you have both wild type and mutants in one test tube or in one plate. So if you have wild type and mutants, then let's say we have, this is a mutant, let's say we also have a purple mutant, oh that's not purple, and then let's say we have a light blue mutant, or light blue is our wild type, so sorry. Okay, and let's say that this is gene AB, and the pink mutant has its problem here, and then the this mutant has its problem here. So say these are all in one test tube. Let's say they're plated high on K. Okay, so we know because the pink and this purple color mutant have the mutation in the same gene, we know that they cannot complement. So let's write that down. We know this mutant and this mutant cannot complement. But we also have the wild type here. So because we have the wild type can complement with either one of these because it has a working B. So because of that, even though our original mutants can't complement, our mutants times wild type can complement. And so this is a key thing to understand, is that while mutants, these two mutants could not complement, because you have another thing in the test tube or on the plate, you actually can have a complementation event occurring. And so you have to be careful when you're going through these experiments to just pay attention to what's coming out of your lysate each time and what that means and whether or not you can complement, recombine, etc. Okay, I hope this helped, guys. Bye.